Okay, let's just get started. Hello everyone. Welcome to our today's Zoom seminar. It's absolutely my pleasure to welcome our today's speaker, Carrie Lucas, from Yale. So, Carrie currently is associate professor of immunobiology in Yale. I met Carrie. I think I should met Carrie long when I joined NIH, but I think we just have a little overlap. When you started, I probably also start same time in NIH. But I met Carrie last year in Cold Spring Harbor. The talk was really fabulous. I was so impressed, so I decided to invite Carrie to on this platform to give us the introduction of her science. So Carrie completed her undergraduate study in University of North Carolina in Chapel Hill, and then continue finishing her PhD in Harvard University, and then she did her postdoc training in NIH, actually in NIARD with Michael Linato. And then from 2016, Carrie started her independent research program as assistant professor in immunobiology from Yale School of Medicine. And then 2022, she was promoted as associate professor. So the work from Carrie's lab actually very much devoted to discovering new and translational relevant principles of immunology by defining and studying human genetic immune disorders, and then combining human genomics in vitro studies using primary patient cells and in vivo mouse modeling. Carrie's team actually looking for to gain incisive basic and translational insight, starting with a patient. Her lab mainly focused on primary immunodeficiency, has largely centered around PI3 kinase signaling and disease mechanism and treatment in patients with a mutation in PI3 subunits, and including the active PI3 delta syndrome, which including APDS1 and NPDS2, and also inactivate PI3 gamma syndrome, IPGS. More recently, her lab has added an emphasis on studying inflammatory diseases, including COVID-SARS-associated multi-system inflammatory syndrome in children, and also vaccine-associated myocarditis. A major currently effort centered on new biology illuminated by monogenic autoinflammatory disease in the Lucas lab disease, and then her lab actually named the disease deficiency in ELL4, X-linked DX. So each of the research projects start with a initial focus on dissecting pediatric immune disease and aim to leverage that knowledge for new therapies, including precision medicine approaches in monogenic disease, which has currently recently been achieved with FDA approval of targeted therapy in APDS. The rare disease study uniquely enabled to elucidation of in-depth mechanistic insight into human immunology, providing translational knowledge to improve understanding and treatment of a broader set of common disease with immune involvement. So with different achievement, Carrie really actually being rewarded with numerous awards, which I'm not going to list all of them, but just a few examples like 2008 to 2018 to 2019, Folk Scholar from Yale, and then 2020 she was awarded as ICIS Young Investigator Award. And then consecutively 5 years from 2023 to 2028, she's awarded as Endo Faculty Fellow from Yale. So it's really a pleasure to have you on our platform, and we are very much looking forward to your talk.
Thank you so much, Tuan. Very kind introduction. I'm really delighted to have the opportunity to present to you all today. I welcome questions if people want to interrupt along the way, but also we'll save time for questions at the end. So as Tuan mentioned, my lab is focused on learning immunobiology, fundamental biology, really, directly from patients. That's our objective and our passion in the lab. So I'm going to tell you about the big picture about inborn errors of immunity, this class of rare diseases. And then I'll highlight a couple of the disorders we've been excited about and have arisen from our work in PI3 kinase that started when I was a postdoc at NIH. Briefly, disclosures, I'm a consultant for farming healthcare and have grant support from Ono Pharma. So big picture, what are monogenic diseases? And, I always like to address this question because some people will ask me, well, Carrie, these are rare diseases. Why do you want to study rare diseases? Don't you want to study something more common that affects a larger population of patients? And my answer is that these rare diseases are unique in that we can definitively determine their cause. So a single gene defect driving a immune phenotype allows us to put our finger on mechanism. And then from there, we can leverage mouse models and in vitro studies to really understand how a particular gene defect is leading to a clinical phenotype. And that new understanding is really what we're excited to contribute through our work. And so what we do, and many in the field of inborn errors of immunity do, is cast a really wide net and look for very rare patients. So these little sort of red people here are meant to symbolize rare patient populations across the world who present with severe or unusual phenotypes that raise the hypothesis for their physicians and for research labs like ours that perhaps this disease, usually early onset disease, would be caused by a single gene defect. And so this approach enables us to take an unbiased method to analyze the trios, so the mom, dad, and affected patient, starting again with an organismal or a clinical phenotype. This involves lots of very productive collaborations with physicians around the world who are experts on the disease that their patient has, and we work very closely with our clinical collaborators. And the goal then is to go from this organismal phenotype to drill into cellular phenotypes in vitro with primary patient cells, as well as molecular phenotypes where we can define that gene defect and understand the circuitry that's disrupted by the mutation. And so just to set the stage and place this, many of us think about genetic contributions to various diseases in the immune system. And if we think about monogenic diseases, here on the y-axis, this is effect size of any given gene variant. And on the x-axis here is allele frequency. So many people are accustomed to thinking about, for example, genome-wide association studies or GWAS studies. This is looking for more common variants in common diseases. So things like taking large cohorts of type 1 diabetes patients or multiple sclerosis patients and trying to understand what variants they share. What do they have in common? And these kinds of variants generally have a small effect size and are more common. For inborn errors of immunity, we're really interested here in the top left, where there's a large effect size of a single gene variant, usually quite deleterious to the function of that protein. And it's rare in the general population. And there's been really an explosion. I don't have time to sort of go through some of the tools that have revolutionized this field, but it's becoming increasingly more tractable to identify single gene defects because of bioinformatics advancements, of course, the cost of sequencing going down, and large databases such as Nomad that have collections of now over 800,000, quote, healthy human genomes or exomes that really give us the power to define what is rare and what is common in the general population. So for inborn errors of immunity, those monogenic single gene defects that cause an immune disease, we're thinking about this category where the gene variant is actually causal, not just sort of associated or contributing, but actually drives the disease, highly penetrant disease. So we kind of think of this as, many of us use CRISPR as a tool to screen for interesting pathways. And we think of this approach as nature's screen, that where nature does the screen for us, these patients show up to their physicians because they've got striking phenotypes, and then we can pursue these cellular and molecular mechanisms. A few observations from the field more broadly. We know that inborn errors of immunity can be caused by both loss and gain of function mutations. Historically, one would think of primarily loss of function, homozygous loss of function mutations, but with time and more sequencing of patients, we realize there can be heterozygous gain of function mutations that can lead to immune disease. And I'll tell you about one of those briefly in the first part of the talk. We're also, as a field, recognizing that it's not only germline mutations that are present in all cells in the body of these patients, but there can actually be genetic errors of immunity that are caused by somatic mutations. So only a rare population of cells could have acquired a somatic mutation that then drives a severe immune phenotype. And so common examples or recent examples people are quite excited about are things like VEXIS, which is mutation in the ubiquitin pathway, UBA1, or TLR8 gain of function. Those are some examples, and there are others. So this is, I think, on the horizon. More and more people will start to uncover somatic mutations driving genetic errors of immunity. Another observation that is encouraging the field to cast a wide net is that the classical sort of idea that these inborn errors of immunity are so severe that they cause lethality in early life. While that is true in many cases, we're realizing it's not always the case. Sometimes these phenotypes can be relatively mild, not necessarily life-threatening. They still cause illness, but can be monogenic even with relatively mild phenotypes. And many can be de novo, so not present in mom or dad, only present in the child. So we're beginning to kind of move away from this idea of very large pedigrees where there's a family history that generation after generation there have been affected individuals, now to more sort of trio-based exome sequencing where the bar is lower to sequence families and try to discover new monogenic defects. And so to set the stage, when we think of IEIs, that's a kind of broad category. And there are two sort of extremes for inborn errors of immunity. One here is on the left. So this graph is meant to depict frequency in the human population here on the y-axis. And then on the x-axis, this is immune responsiveness. And so in the schematic, there are rare individuals that have very low immune responsiveness, and that leads to infections. So you get normal exposures throughout life to pathogens and opportunistic infections, and these patients can't control them well. These are what we call PID patients, or primary immunodeficiency. And more, there's a lot emerging by many labs around the world. We're also interested in sort of both sides of this curve. But here on the right, it's a separate category where there's a very high level of immune responsiveness. And here you have single gene defects that drive the immune system to be sort of hyperactive. And because they're hyperactive, they drive tissue immunopathology. So not necessarily infection, but rather more autoinflammatory or autoimmune kinds of conditions that are single gene, driven by single genes. And we call those PIRDs, or primary immune regulatory disorders. So both sides of the spectrum are quite interesting and can teach us things, again, in an unbiased way. So we don't know what gene we're going to find at the outset when you've got the pedigree, but it can teach us and give us leads to interesting pathways. The other answer I give people when they ask, why do you study rare diseases, is also that collectively, these diseases are not that rare. So each individual monogenic defect is rare, but when you put them together, it affects millions of people. And there are over 550 genetic errors of immunity that have now been reported. So this is the most recent tabulation of all the IEIs from the new journal that we've got, the Journal of Human Immunity. Just came out recently, and you can see we're over 550 now that the field has identified, really with an explosion around the time next-gen sequencing became accessible. And what's also exciting is, I think, although many have been found, we still have a lot more to find, and we're able to identify these patients not only because they seek care from clinical immunologists for their infections or rheumatologists for their inflammation, but also a wide range of other kinds of clinical phenotypes. So you could find patients from gastroenterologists who refer patients, et cetera. So a wide range of immune-related clinical presentations. And the reason we want to do this is to give new diagnoses to patients, and hopefully those diagnoses can help with precision treatments. And so I'll tell you a bit about an example of prior work that we've contributed to of this activated PI3 kinase delta syndrome, in which there was a tailored therapy that arose because the genetic etiology was understood. And then the hope is we can, because we're learning new biology, we can apply those concepts to more common diseases to help even more patients. We've, I think, covered most of these. There are a few examples where it's sort of exciting to think about monogenics as a way to identify new therapeutic targets. For example, people will often refer to BTK deficiency. So this was the first primary immunodeficiency discovered back in 1952, described by Dr. Bruton. And now BTK is being targeted with small molecule inhibitors as a therapy against B-cell malignancies and other B-cell pathologies. So learning directly from humans that BTK is important provides insights that then again helps with therapeutic targets. So we think of each of these single gene defects as a piece of the puzzle of how the human immune system works. And one of our goals is also to try to think about them broadly, each one individually, but then also all of them collectively to learn immunology at a systems level. So the motivation for me to initially really get excited about this field relates to stories like this one. So some of you may recognize this is the famous boy in the bubble, David Vetter, and this is a boy who was born to a family that had a prior son who died of infection in infancy. And they knew there was a high probability that if they had another son, that son too would suffer from severe infections. And so as soon as he was born, he was put into a bubble. And this was also, there are movies, John Travolta movies and others that depict the story of David Vetter. And it was sort of captivated the attention of the public. And he lived 1971 to 1984. He was treated with a bone marrow transplant. Unfortunately, that was EBV positive. So he passed away from post-transplant lymphoproliferative disease. And he has what's now known as severe combined immunodeficiency, or SCID, this boy in the bubble syndrome. And there are multiple genetic causes of SCID. In his case, it was the X-linked cause, IL-2RG, which that genetic basis was not solved until about 10 years after his death. But this really illustrated and got me excited about this field in the early days, because this is a good example of where at the time the field really only understood the tip of the iceberg. So the gene name, as I mentioned on the last slide, was IL-2 receptor gamma, IL-2RG. It was known to be a chain for the IL-2 receptor. But at that time, in the sort of early mid-90s, knockout mice were coming around as well. And people had made IL-2 knockout mice. And in that case, T cells were still present. And there was some sort of puzzle about why, when you knock out the receptor gamma chain, do you get a different phenotype than when you knock out IL-2? And of course, now we know that IL-2RG is not only a receptor chain for IL-2, but also for several other cytokines listed here. So this is now what we call the common gamma chain. But starting with human diseases helped to raise the questions that led now to that discovery that this is a receptor chain important for multiple cytokines. And this is why when you have mutations in these SCID patients, they're very severely immunodeficient, because all of these cytokines lack normal function. So we were really motivated to kind of tackle this and try to see if we could find more diseases like this boy in the bubble disease that would teach us new knowledge and help us understand more than just the tip of the iceberg. So what I'm mostly going to talk about today are immunodeficiencies. We have a lot of interest also in the PIRDs, the primary immune regulatory disorders, but for today I'll focus on antibody deficiencies. And one of the reasons antibody deficiencies are important is because they really make up the largest fraction of the variety of different types of primary immunodeficiencies that patients can have. So if you look here in this pie chart from IUIS, you can see in blue, these are antibody deficiencies, and then plus the purple part, which are combined, both cellular, so T and B cell deficiencies. SCID is an example of that. So the purple plus the blue is sort of the fraction of the primary immunodeficiencies that include antibody defects. So it's a really significant proportion of immunodeficiency. And you can sort of understand why that might be if you think about the biology required to produce high affinity antibodies. So this is just a schematic from a nice review article from Gabriel Victoria and Michel Nussenzweig, just to highlight for you the complexity of the germinal center reaction. So we know not only do B cells need to develop normally in the bone marrow and mature properly, but they also, when encountering T-dependent antigens, need to go through a very intricate process of entering a germinal center where they can get T cell help, go into the dark zone then and undergo somatic hypermutation where they can then compete and test their affinity. So this is a very complex biology. They then differentiate into memory B cells and also plasma cells that can produce high affinity antibody to maintain serum levels. So many steps along the way could go wrong. And I think this is why we see such a large fraction of primary immunodeficiencies having antibody defects as part of their picture. So it's an important problem, and one we think the field will continue to identify and elucidate biology of the antibody response by studying this class of primary immunodeficiencies. So for today, I want to talk about two diseases, and I'll start with an overview of the disease that Chuan mentioned, this PI3 kinase delta gain of function, what is now known as activated PI3 kinase delta syndrome, or APDS, which I was fortunate to work on as part of a collaborative group at NIH, and continue to contribute and think about ways we can learn from PI3 kinase delta mutations. And then I'll spend most of the time on new studies on another PI3 kinase gamma, and what it's teaching us about the B cell response. So just to frame sort of what we're thinking about and summarize a little bit of what I mentioned already. So the field of inborn errors of immunity is this broader circle. And within that, there's a category of primary immunodeficiencies, many of which include antibody defects. And we're going to talk about two of those diseases here, APDS and IPGS. The APDS story is one that started when I was a postdoc, and I'll briefly describe for you the clinical phenotype and what's been shown. But one reason I like to highlight this is it's a rare case. There are other examples, but this is a rare case and an exciting example in which immunodeficient patients were found to have heterozygous mutations that cause gain of function in PI3 kinase. And it's relatively unusual to have a very straightforward way to treat patients that have a single gene defect. And in this case, because you have hyperactivation of a kinase, one could pretty logically test the hypothesis that they'll respond well to inhibitors of that kinase. And so this is a story where the genetic etiology was published in 2013, 2014, and about 10 years later, so in March of 2023, there was FDA approval of a specific small molecule inhibitor that is now helping these patients. This disease affects a relatively large number of patients. And so I'll walk you through the highlights of that story, and then move on to this related PI3 kinase gamma, and tell you about the elements of plasma cell differentiation that we're excited to have recently discovered. So to orient you about the pathway, many of you will be familiar with PI3 kinase signaling. It's a very conserved signaling pathway involved in lots of cell growth and proliferation signals. In the case of the immune system, there are two that are relatively specifically expressed in leukocytes. And those are PI3 kinase delta, the first one we'll talk about. And this responds to receptor tyrosine kinase signals. So when you have phosphotyrosine, the SH2 domains here of the P85 regulatory subunit will bring that kinase complex to the cytoplasmic tail of the phosphorylated receptor and initiate downstream signaling, where PIP3 second messenger is produced, and that allows for AKT and mTOR activation that promotes metabolism, cell growth, and proliferation. The second one that's pretty immune cell specific is PI3 kinase gamma, which traditionally has been thought of as primarily a myeloid PI3 kinase because it's expressed at highest levels in myeloid cells. And instead of responding to tyrosine phosphorylation, PI3 kinase gamma is activated by G beta gamma, so downstream of GPCR signaling. And in the early days was studied in the context of chemokine receptor signaling and chemotaxis in neutrophils and other myeloid cell types. So these are the two kinases we're going to talk about. They both lead to production of PIP3 and very similar downstream signaling pathways through AKT and mTOR, but they're triggered by different signals, and they're expressed and sort of thought to have dominant roles in different subsets, where delta is more in lymphocytes, and previously gamma was thought to be primarily in myeloid. So PI3 kinase delta is the first one we're going to start with. This is our early paper. I mentioned I put this here so you can see all of the co-authors, very collaborative work where many continue to work on mechanisms that we can uncover from APDS, including Stu Tange, Pam Schwartzberg. And then we had colleagues publishing it at just a bit ahead of us who also continue. So Klaas Aukenhag, for example, has done important work in mouse models of APDS, and we continue to learn more about how PI3 kinase regulation affects the immune system through this work. And so this was our original report where you can see this is the catalytic subunit of P110 delta. And these are the sites of the mutation. So multiple different areas across the catalytic subunit. Interestingly, there were multiple patients with this C-terminal mutation, and this has turned out to be the most common mutation now across hundreds of APDS patients that have been reported. And if you look at how this is hyperactivating, most of these mutations affect the interface between the catalytic subunit and the P85 regulatory subunit, such that the regulatory subunit can't inhibit the catalytic subunit. And this is why it's a gain of function. These patients have one copy of the mutant allele. Their other allele is wild type, and it's sufficient to drive this disease. There's also, shortly thereafter, we and others from Sven Kracker's lab, this was while I was in Mike Leonardo's lab, found mutations in that binding partner. So P110 delta is the catalytic subunit, P85 is the regulatory subunit. And you can see we started with 3 families. Now there have been many described. And here what happens is there's a skipping of an exon, and it actually produces an in-frame transcript. So you get a truncated form of the regulatory subunit here, P85, so that it can still bind to the catalytic subunit, but it doesn't inhibit it properly. So again, in both cases, the mechanism results in hyperactivation, augmented PI3 kinase signaling. So you can sort of think about it as a disease of too much of a good thing. This is just one. There are multiple examples of clinical summaries where our colleagues across the field have sort of summarized what do APDS patients present with, why do they come to their physician. And what's most common is recurrent respiratory tract infections, sinopulmonary infections. And because of the recurrent sinopulmonary infections, they end up with bronchiectasis, which is sort of mechanical damage in the lung. And when physicians see a picture like this of recurrent sinopulmonary infections with encapsulated bacteria, it really makes one think that there are antibody defects. And indeed, I'll show you more shortly, APDS results in poor B cell development and bad antibody responses, so low IgGs. So recurrent infections, sinopulmonary infections, there's also lymphadenopathy, so swollen lymph nodes, swollen spleen, and difficulty controlling herpes viruses, which relates to their T cell phenotypes. When PI3 kinase is high, they hyperproliferate and senesce and are less effective at controlling these latent viruses. So because much of this is published, and I want to save time for us to get into the PI3 kinase gamma side of things, I wanted to summarize some of the immunology lessons from APDS from this review article, where we know in health that there's really a balance. PI3 kinase delta needs to be able to at times be elevated, and other times during the course of lymphocyte development and lymphocyte activation be tuned down. And when you have a mutation that occurs in APDS that drives PI3 kinase delta signaling to be constitutively too high, we have disruptions in these pathways. So as I mentioned, there's an increase in T cell senescence and T cell death, an increase in transitional B cells, so B cells aren't maturing properly, and there are defects in class switch recombination and somatic hypermutation. APDS patients have an increased susceptibility to B cell lymphomas and, as mentioned, infections and lymphadenopathy. There are also very rare cases of patients who have loss of function mutations in these PI3 kinase subunits, and they also have immunodeficiency. So whether you turn it up, lock it up at a hyperactive state or an inactive state, it results in immune phenotypes. And there's been a lot of work to dissect how having elevated PI3 kinase delta signaling disrupts B cell development and leads to antibody defects and poor T cell help in germinal centers. So hopefully this will continue to yield new insights that are relevant, not only for APDS, but related diseases with lymphadenopathy and IgG defects. I mentioned briefly the tailored therapy, so precision medicine approach, again, because there are small molecule inhibitors. In this case, it was originally developed by Novartis and had been tested in other immune disease contexts and was sort of a perfect match and led to trials. There were other PI3 kinase delta inhibitors also tested that had different routes of administration, but this is the one that is now FDA approved. And so if you're interested, you can see these papers here and look at the studies. I'll show you just a little bit of data where this leniolisib is restoring some of the abnormal features in APDS. So this is showing you naive B cells at baseline and then at various time points after treatment with the small molecule inhibitor. The gray area is the normal range. And so you can see it's restoring naive B cells back to normal levels and reducing those transitional B cells. So the placebo group remains high and the treated group normalizes, as well as other serum factors such as CXCL13 gets normalized. So this is encouraging and exciting and has yielded benefit for APDS patients. And so this summarizes for you that it's a sort of rare example where overactivation of a pathway can be precisely targeted and lead to benefits. There are cancer contexts in which other PI3 kinase delta inhibitors have been used to try to help in B-cell malignancies, and that can lead to things like colitis. But thankfully, there's not a strong signal for that in APDS patients, likely because they start already with an elevated level of PI3 kinase signaling. So their dosing can be optimized for on-target effects without the downsides of over-inhibition of PI3 kinase delta. So that then, of course, led to a love of PI3 kinases. And because we knew that it wasn't just PI3 kinase delta that is expressed primarily in immune cells, but also PI3 kinase gamma, we were really excited to continue to work with colleagues and try to identify patients who have mutations in this other kinase, PI3 kinase gamma. And so that's what I'll tell you about in my remaining time. So we first reported PI3 kinase gamma deficiency. So now this is not gain of function like we had for delta, but biallelic. It's a compound heterozygous mutation that results in loss of PI3 kinase gamma. So this is sort of a human knockout where there are two elements of the patient phenotype. One is inflammatory. So in the barrier tissues, in particular the lung and the gut, there's lymphocytic pneumonitis and colitis, which in our early 2019 paper, we studied the myeloid cells, macrophages in particular, because PI3 kinase gamma had been implicated as being a sort of checkpoint in myeloid cells. And so either a small molecule inhibitor or genetic deletion of PI3 kinase gamma in macrophages, when you stimulate them then with pattern recognition receptor ligands, they were hyper inflammatory. And so in our initial report, that's what we focused on and tried to understand if that contributed to the patient phenotype. We did this also using PI3 kinase gamma knockout mice that we got from Matthias Wyman in Switzerland. So that sort of fit with prior knowledge. As I mentioned, PI3 kinase gamma is one that's known to be expressed at high levels in myeloid cells. But the second element of this patient phenotype really surprised us, and that is this pattern of recurrent sinopulmonary infections with encapsulated bacteria and antibody defects. And so this patient required immunoglobulin supplementation to help protect from infections and some autoimmune type conditions that can happen in CVID and related antibody deficiencies. And that really was a surprise because PI3 kinase gamma really was thought of as primarily myeloid, and why would this lead then to a B cell deficiency? And so we were intrigued and wanted to dissect this further. On the right here, I'm showing you these are the patient alleles, and you can see in expanded T cell blasts from the patient, there's really no P110 gamma protein. This is her serum immunoglobulin levels, and so you can see quite low prior to Ig supplementation, and then after getting Ig supplementation, of course, she goes back up into normal levels and is protected from recurrent infections. And so one of the early experiments we did, sort of motivated by some prior work by labs such as Dave Massapus' lab, we really are interested in dissecting biology relevant for human immunology, of course, because we start with humans, but the powerful mouse immunology tools really allow for mechanistic dissection. So in my lab, we really like to pair them. And so we wanted to better recapitulate the human phenotype. And there had been 20 years of work with PI3 kinase gamma knockout mice, and nobody had reported or sort of discovered this antibody deficiency or the lymphocytic pneumonitis and colitis. And so one of the early things we did was a co-housing experiment. We hypothesized that simply maybe the reason that the mouse phenotype is different from the human phenotype is because they don't have exposures to normal microbiota that mature the immune cells in important ways. And so we carried out a series of experiments in which we allowed for transfer of healthy but very diverse microbiota from mice we purchased from a pet store. And they then were transferred into the laboratory C57 Black 6 mice, either wild type or PI3 kinase gamma knockout littermate controls. And we let them regain in weight and find their new homeostasis and assessed various parameters. We were able to see lymphocytic infiltration of barrier tissue was increased in the knockouts with this approach. But importantly, we also found broad reductions in various classes, IgG subtypes. And so in red, you can see are the knockout compared to wild type. These are all, quote, dirtied mice. And so all of their immunoglobulin levels have been boosted compared to prior to co-housing. But you can see they're boosted to less of an extent in the knockouts compared to the wild type. So early suggestion that we can use mouse models to dissect this biology of the antibody defect in PI3 kinase gamma deficiency. So Stephen Lanahan was a talented PhD student who bravely tackled this new project and wanted to use very controlled systems. And so using NP-OVA immunizations, first recapitulated this antibody defect. So this is looking at IgG1, either total IgG1 or high affinity IgG1 in controls versus PI3 kinase gamma knockout. And we saw striking reductions in the ability to produce NP-specific IgG. I don't have time to show you, but he did very careful analyses of B cell development, IgM responses, IgA responses. These all looked normal. It was an IgG defect. And so what I've shown you so far is that the human gene discovery has taught us that PI3 kinase gamma is required for human, and now we also found mouse T-dependent antibody responses, leading us to a few next questions. So firstly, at which stage of the response does PI3 kinase gamma act? So we talked about the complex biology of producing high affinity IgGs. So where in that response is PI3 kinase gamma important, and in which cell types? So a few pieces of negative data. We thought, well, PI3 kinase generally and AKT-mTOR pathways are really important in cell growth and proliferation. So we hypothesized maybe it's important for B cell proliferation, but really couldn't detect a defect in proliferation with various types of stimulation. And this isn't surprising because these cells still express PI3 kinase delta. And delta is known to be the dominant PI3 kinase in lymphocytes and is important for things like B cell development and proliferation. So this is likely why this is normal, even when PI3 kinase gamma is deficient. This is after sheep red blood cell immunization in het controls or PI3 kinase gamma knockout mice, and we could see normal formation of germinal centers. This is staining for GL7. So it wasn't that the germinal center response couldn't happen at all. We also worked with Steve Kleinstein and Gisela Gabernet to look by single cell at controls versus knockouts at GCB cells to look at class switch, and this looked normal, no defects in class switch recombination. So germinal centers can form, B cells can proliferate, class switching can happen, and mutation frequency was not different. So the germinal center response itself by these measures was intact, but was really insightful for us was when Stephen, after immunizing animals and boosting them at a late time point after immunization, sorted out germinal center B cells by these markers you see here, and did bulk RNA sequencing to get an in-depth transcriptome assessment. And you can see what stands out on the right here, these are 4 knockouts, and on the left are 3 controls. And these are various genes that are important for activated and GC B cells. And you can see they're elevated in the knockout. Again, this is 28 days after immunization, and you still are seeing active germinal center response, as opposed to in the wild type animals, you're seeing a conversion towards pre-antibody secreting cells, or ASCs. So basically, the germinal center B cells are able to become more plasma cells in the wild types, but not in the knockouts. So a few of the key plasma cell genes you can see, PRDM1, IRF4, XBP1, for example, these are all low in PI3 kinase gamma knockouts. So this was the first sort of hint that what was perhaps defective when you lacked PI3 kinase gamma was the ability of an activated B cell, which could proliferate normally and make a germinal center response, but without PI3 kinase gamma, it couldn't become an ASC, a long-lived plasma cell that's important for producing those IgGs that we're detecting in the serum. And if we did gene set enrichment analysis, we could see, as you would expect, that mTOR pathways, genes associated with mTOR signaling are lower, as is the unfolded protein response, which is really important as plasma cells produce so many proteins and have a lot of ER stress and unfolded protein response that they have to deal with in order to produce IgGs at such a rate. So this kind of raised the idea that maybe PI3 kinase gamma plays a particular role as activated B cells become plasma cells, or ASCs, antibody secreting cells. And we know PI3 kinase has been implicated in promoting things like IRF4, which is important in plasma cell differentiation, and wanted to further interrogate if this could be why humans and mice deficient in PI3 kinase gamma have poor antibody responses. And just going back to the basics with expression levels, one of the observations Stephen made is shown here. So on the left is showing you PIK3CG, this is PI3 kinase gamma, and on the right, PIK3CD, or PI3 kinase delta. And as expected, we knew that gamma is at high levels in myeloid cells. So you see the monocytes have high expression of gamma. But interestingly, in this data set, there were plasmablasts as well. This is from human. And you can see there's almost equivalent level of expression of PI3 kinase gamma at the plasmablast stage. And at the same stage, there was a reduction in delta. And if he looked in mouse publicly available data sets as well, or in his own RNA-seq data sets, he could see that germinal center B cells, follicular B cells, germinal center B cells have expressed both. And of course, across all of these lineages, there's expression of both of them, but the relative abundance changes as a GCB cell becomes a plasmablast or a plasma cell. They're expressing relatively higher levels of PIK3CG and relatively lower levels of PI3 kinase delta. And we think that this is important because an IgG B cell, an IgG germinal center B cell, as it's becoming a plasma cell, downregulates its surface BCR. And we know surface BCR is tied to PI3 kinase delta. And a cell that doesn't have that receptor on the surface anymore may now depend more on PI3 kinase gamma and signals that converge on gamma in order to transduce signals to AKT and mTOR. So this kind of molecular switch towards PI3 kinase gamma function as a B cell becomes a plasma cell was intriguing and made us wonder which cell type requires PI3 kinase gamma, because these data would make us think it would be intrinsic within the B cell. And so Stephen did a lot of crossing of various mouse lines and his NP-OVA immunization approach. And you can see MB1-CRE, this is now specific deletion of PIK3CG floxed allele from B cells. And you can compare the dotted line here is the total knockout. So the whole body knockout would be at about this level. And knocking it out specifically in B cells led to a phenotype nearly as severe as the total knockout. Whereas knocking it out in other cell types had no effect or a smaller effect in the case of CD4-CRE. So this was great and suggested that it is actually a B cell intrinsic requirement. And in these same flux animals, he could also look at ASCs by ELISpot, so looking at production IgG producing cells, and they were reduced. So this switch from an activated B cell into a plasma cell still is defective when PI3 kinase gamma is specifically deleted from B cells. So we can answer these two questions now, that the stage where it's important is as this activated B cell becomes an antibody secreting cell, and it's important in B cells themselves. So then that would lead us to the prediction that disrupting this kinase, PI3 kinase gamma, in B cells, even after activation, since we think it's that late stage where it's playing a role, that should also be sufficient to diminish the antibody response. And we have tools to test that using C-gamma-1 Cree. So this Cree will only turn on in a B cell after activation. So the B cell develops normally, it has PI3 kinase gamma, it can become activated and then delete PI3 kinase gamma. So it will be gone at the stage when an activated B cell becomes a plasma cell, and we can recapitulate the IgG defect even in those animals. Similarly, we thought we could use small molecule inhibitors. So I touched on in the first story that there are inhibitors of delta that have helped APDS patients. There are also specific inhibitors getting better and better at being specific for PI3 kinase gamma. And so we wanted to use this now as a tool to see, can we recapitulate with a pharmacologic agent, again, at a later time point, can we recapitulate the reduction in antibody response? And so here we immunized the mice, and then between days 3 and 12 administered this IPI 549 small molecule PI3 kinase gamma inhibitor by oral gavage. And you can see unimmunized versus vehicle. This is again ELISpot looking at IgG producing cells. And when we administered the small molecule drug against PI3 kinase gamma, there's a significant reduction in antibody secreting cells. And so that further confirms that you can initiate a response and then blunt it if B cells can't utilize this kinase when they need to become antibody secreting cells. And we wanted to circle back to human biology, and we're lucky to team up with a colleague and collaborator, Neil Romberg at CHOP, who has set up tonsillar organoid models following protocols similar to those from Mark Davis's lab. And he used this model in which you have human tonsil and titrated in PI3 kinase gamma inhibitor at the doses shown here. And our prediction was we could, in human, as we saw in mice, show that this inhibitor can block the ability of activated B cells to become plasma cells. And so here in this red box is the gating. Here he's using IgD and CD38, also CD138 markers show similar things. But you can see as you titrate up the small molecule inhibitor, we're seeing fewer and fewer plasmablasts in these tonsillar organoid models. Consistent with the conclusion that it's true in mouse and human biology that B cells require this kinase for that transition. We could also, in addition to the tonsillar organoid model, we could purify human B cells and get them to differentiate through the various cytokines I'm showing for you here into antibody secreting cells. This is showing you a dose titration looking at human pan B cell culture, dependence again on PI3 kinase gamma for their differentiation into plasma cells. And it wasn't just primary stimulation, which many of what I've shown you so far is really taking a naive cell and activating it through immunization or in these in vitro ways to become an activated B cell and then an antibody secreting cell. Importantly, it's also true for secondary plasma cells where you take a memory cell, so a B cell that's already experienced antigen, it's acquired a memory phenotype, and we can sort those from human using the CD27 marker and ask those memory B cells to differentiate into plasma cells just through stimulating TLR7. That's sufficient to get differentiation into plasma cells. And even in that context, we see a dose-dependent reduction in plasma cell differentiation when we inhibit PI3 kinase gamma. And one piece of the mechanism we think, I showed you prior gene expression data and in the schematics really pointed out for you that IRF4 is a critical transcription factor promoting plasma cell differentiation. And in the situations in which we've inhibited PI3 kinase gamma, we see less induction of IRF4. So potentially implicating PI3 kinase gamma as being important in transducing signals that allow an activated B cell to upregulate this important transcription factor for plasma cell differentiation. So you can read in more depth. This was published late last year. We can, if you're interested, you can see more details there. And we have ongoing work. Really, there are several unknowns that we're excited to tackle, and those include what are the upstream receptors. So I told you that PI3 kinase gamma is activated by GPCRs. but we don't know which GPCR, or if it is a GPCR, can also be activated by RAS-related signals and others. And so we're currently hard at work trying to find what are the upstream receptors and which downstream effectors of the kinase are most important for this transition of an activated B cell into an ASC. And in terms of application, although an inhibitor, of course, would not be helpful for a human that has genetic deficiency, this rare disease that we started with, but there are lots of other disease contexts in which there are pathogenic antibodies that one would like to reduce. So, for example, in autoimmune disorders driven by antibodies, or maybe even in plasma cell neoplasms, would it be helpful to specifically inhibit PI3 kinase gamma in those contexts and blunt the antibody response or plasma cell cancer itself? And so to synthesize what I've told you so far, we've started with a human disease that has led us to a series of investigations about how PI3 kinase gamma supports the antibody response. I didn't have time to show you everything, but we see that there's no role for PI3 kinase gamma in T cell independent antibody responses, but in T cell dependent responses within the B cell, it's important as a germinal center B cell becomes a plasma cell. And without it, either genetically or pharmacologically, we see reductions in antigen-specific antibody responses. We think that this helps to explain this molecular switch that we're observing. So the expression patterns fit with the timing for when we see PI3 kinase gamma is important. So as an activated B cell becomes an ASC, it's downregulating delta and internalizing its IgG B cell BCR, and therefore relying more on PI3 kinase gamma at that stage. And PI3 kinase gamma then promotes IRF4, which is important for the ASC differentiation. And this is true whether this is a naive B cell that's been activated for the initial activation, and also true with memory B cells in which the stimulation is just TLR stim. So whether it's CD40 and BCR or TLR stim, this is still required for differentiation into plasma cells, which we think is giving us a clue into some of the converging signaling pathways that would be driving this biology. I showed you a gene set enrichment analysis that suggested it's important for gene expression related to metabolism and the unfolded protein response, which we're dissecting now further. And we're busy at work also trying to understand the implications in disease contexts such as autoantibody-driven autoimmunity. So that's the two PI3 kinase stories, and hopefully I've convinced you that this field of studying inborn errors of immunity can not only help give diagnoses to patients, but teach us some new biology. So I sort of like to think about this as, because it's an unbiased approach, most science, we kind of look under the lamppost where we know there's some prior knowledge and it makes sense to test a hypothesis sort of not too far from what it is that we already know. Whereas these single gene defects allow us to shine new light on biology that you couldn't have predicted otherwise. And so these rare phenotypes function as a flashlight to help us illuminate this new biology. And so I'll just take a minute to acknowledge the people in the lab who did the work, and really start by also thanking the patients, their families, and many clinicians and colleagues that we have the privilege to collaborate with. Of course, this research couldn't be done without them. These are current lab members, and I've underlined here the primary members for the PI3 kinase gamma work that I mentioned, with Stephen here being the main driver. We collaborate closely with colleagues in Pediatric Genomics Discovery Program for our sequencing, and I mentioned some of our clinical colleagues along the way, including Kanedi Rao and Peter Mustillo, Roshini Abraham, who have helped care for PI3 kinase gamma patients, Matthias Weimann for the PI3 kinase gamma knockout mice, and our sources of funding. And I would be happy to take questions.